Hello, I am Carolina González, and today I will show you our results about the comparison of different phylogenetic methods for microbial species delimitation using Acidithiobacillia class as a model. Our laboratory focused in the research of the extinct acidophile, which are defined as a microorganism with a pH optimum of growth less than 3. The acidophile distribute in different clade of the tree of life, represented by the red star in this adaptation of the tree of life. And today we will focus in the Acidithiobacilliaceae family. So when we decide to study our acidophilic branch, we can identify two cases. The case A, where the acidophile groups in one clade and arose from a neutrophile, or the case B, where the acidophile gives rise to a neutrophile. In this case, neutrophile means something different to acidophile, neutral or higher pH. So to identify if we have the case A or B, we construct a phylogenetic trees using different methods. Single locus, where the gold standard method is the 16S ribosomal marker. Multi-locus, when we use several markers. Or whole genome analysis, when we compare the composition of the genomes. We develop all these methods for the Acidithiobacillia class, and according to our result and the literature, we confirm the Acidithiobacillia class separate in two main clades. One of them grouping the neutrophile, and the second all the acidophile represented by red. So we obtain the case A, where the acidophile give a rise from neutrophile. Also, the sister clade gamma and beta protobacteria and the ancestor clade Zeta protobacteria include neutrophile. So this allows to propose that the ancestor of the Acidithiobacillia was a neutrophile, confirming the case A. Then we decide to go deeper in the analysis and identify how many species are included in the Acidithiobacillia class. For that, we use the gold standard, which corresponds to the comparison of the 16S ribosomal sequence identity. According to the cutoff proposed for microbial species definition in 1997, if two sequences have at least 97% of the identity correspond to the same species. We apply this cutoff to the Acidithiobacillia class and identify that the class have two species. If we use the new cutoff that is proposed to be 98.7%, we obtain 5 species. According to our analysis and the literature, we know that we require at least a 99.7% of identity to obtain 10 A groups in this case. All this using 36 genomes from the Acidithiobacillia class as examples. So if we keep using the actual cutoff value used for species definition, we obtain 5 groups. However, if we use the new method as multi-locus sequence phylogenetic tree and whole genome analysis, we can increase the variability, and we know that we have at least A group in the Acidithiobacillia class. This heat map was constructed using the average nucleotide identity. So if two genomes correspond to the same species, have at least a 97% of the shared genomes. So as conclusion and perspective, a robust phylogenetic tree is key and is the first step required for evolutionary research. A 16S ribosomal phylogenetic tree generates a significant number of unsupported associations and allows to incorrect taxonomy and conclusion. Phylogenetic tree of multi-locus markers and whole genome analysis, such as average nucleotide identity, allows to construct a robust phylogenetic tree and obtain correct conclusions. And in the case of the Acidithiobacillia class, or phylogenetic analysis and whole genome analysis propose that the Acidithiobacillia evolved from an ancestral neutrophile, gaining the ability to survive in extreme acidic environment. If you have more questions, you can write me to this email. Thank you for your attention.